Payne West is the leading broker in the Pacific Northwest where Vandals could shop for home insurance, auto insurance, and more. And every new policy purchased supports the U of I Alumni Association. And thanks to Payne West, every attendee tonight is automatically entered into a drawing for a $50 Vandal card, Vandal store gift card. We'll announce the winner at the end, and you have to be present, such as it is, to win. So we'll do that a little bit later. It's now my pleasure to introduce two amazing Vandals who are doing really, really important work. Pavitra Roy Shithuri is an acting instructor at the University of Washington and a research associate at the Fred Hutchinson Cancer Research Center in Seattle. She has a Master's of Science degree in biomedical engineering and earned her PhD in bioinformatics and computational biology from the University of Idaho in 2013. She currently leads the genomics team within the UI Virology Lab, which has been heavily involved in clinical testing of SARS-CoV-2. She will describe how UW virology efforts in sequencing the genome of SARS-CoV-2 have contributed to our understanding of the pathogen. Dr. Colin Fields graduated from U of I in 2004, and in 2010, he graduated from Ben Gurion University of the Negev Medical School for International Health. He is a family physician at Kaiser Permanente, Washington, or KPWA, and he lives in Seattle with his family. In his professional capacities, he is the chief of both the KPWA HIV and PrEP program and the KPWA Gender Health Program. In addition, he works as a sub-investigator for vaccine clinical studies at the Kaiser Permanente Washington Health Research Institute. It is so great to have you both here. And as I was saying earlier, the timing just could not be better. This is excellent. We have a lot to talk about. But I want to start by asking you how you and your families are doing in the pandemic, navigating all the changes that we've seen. Colin, why don't you go first?
That has been quite the moving target for me.
Of our university.
Since
pleasure from
I started, well, since COVID started, sorry, I think that there's some people who aren't muted.
Yep, everyone could mute, that would be great. And Colin, I'm having trouble hearing you. So if you could either turn up your volume or lean in, that would be great.
Can you hear me now better?
That's better, yes.
Okay. So when COVID started, I was just living here with my husband, but then we ended up adopting our son in May. So things have changed quite a bit for us in terms of COVID life. But I think overall things are going pretty well. Right now I'm in my basement where I often see patients virtually right now, in addition to going to clinic in person sometimes as well. So it's been a lot of changes during this 2020 year.
I would say a new baby, my goodness, that is a big change, life change, especially in this time. So congratulations to both of you. Pavitra, how about you and yours?
Yeah, hi everyone. Thank you all for coming, and thank you for having me. So my involvement with COVID started on February 28th. So that's when we received our first positive sample from the state public health lab, and we wanted to sequence that sample and find the genomic sequence of SARS-CoV-2 in that sample. And that really, that's where I started out. And then March 1st, the day after that, our lab got approved to do clinical testing for SARS-CoV-2. And that, I don't know, you may have seen in the news, and, it was the first in Washington state, and really that set things off. And since we were sort of among the first to have a major outbreak in our states, in the country. So since that time, our lab has tested more than 1 million samples for SARS-CoV-2. And our charter, when we started out, was to sequence every positive sample. And so starting February 28th, that's what we've been doing. And I lead a pretty small team here, because it's literally all hands on deck. Because you can see my office has biorad storage area as well. So, yeah, basically we wanted to sequence every positive sample, and I know we'll talk about that a little bit, but we've just kept that up. We've sequenced more than 5,000 samples so far. We've had a big focus on depositing that data, making it publicly available so that others around the world can use it. So that's kind of been professionally. And then personally, I think we've been similar in many ways to the rest of the world in experiencing lockdowns and things like that. Fortunately, my kid is still in daycare, and daycare has remained. This particular daycare serves Fred Hutch, and so it has remained open, which is so that's been great. With a lot of precautions and things like that. And knock on wood, no positive cases so far in the daycare. So, but yeah, it's been different.
Yeah, it has been different for all of us, for sure. So, Pavitra, I want to start out. Can you tell us a little bit about what coronaviruses are and where they come from? Because COVID-19 is just one of many, right?
Yes. So SARS-CoV-2, which is a virus that causes COVID-19, the disease, is a coronavirus, which is a sort of family of viruses. And these are not, the family of viruses is not new. These coronaviruses have existed for thousands and thousands of years. And there are coronaviruses in many other species, bats, birds, and so on. Human coronaviruses, I think the first human coronavirus was identified probably in the 60s, but it's thought to have been in humans for something like 10,000 years or something like that. So they're not new as a family, but SARS-CoV-2 is a novel virus in the sense that this is the first time that we are seeing this in a human population, and we're still learning a lot about it. But yeah, coronaviruses are a kind of virus. They're called that because of the way they look under the electron microscope. They have a little halo or corona. So that's where the name comes from.
Oh, that's fascinating. I didn't know that. That's really interesting. Okay.
And actually, a lot of the human coronaviruses that exist, the non-SARS ones, there are many that are just seasonal, and they cause fairly mild respiratory disease. So SARS-CoV-2 and its closer relatives like SARS-1 and MERS are different though.
Okay, great. Colin, there are multiple symptoms for COVID-19. What part of the body does it actually attack when a person gets COVID-19?
The usual way that COVID-19, and can you hear me, by the way?
Not very well.
Let me try a different microphone. Maybe
Okay.
Pavitra should take another question, and I'll.
Sure. So we were going to talk about SARS and MERS and influenza. What are the differences?
Yeah, so SARS-CoV-2, SARS-1, and MERS are all coronaviruses. They're different in the clinical side of things as well as the genomic side of things. They're distinct viruses, and they seem to cause somewhat distinct disease as well. So SARS-CoV-1 and MERS have sort of cause more severe disease, for example. They're also genetically quite different, but they are related in that they're part of the same family. Influenza is not a coronavirus. It is a respiratory virus. It's also an RNA virus, which is the material that the nucleate that the virus is made of, its genome is made of. But it's quite different in the way its genome is structured. For example, it mutates a lot faster because it can like shuffle pieces of its genome around, and that's one of the reasons why it's kind of challenging to develop a vaccine for influenza, for seasonal influenza, among other things. And they're also, like I said, different from the clinical end. And I know Colin will chime in better about that, but influenza can also kill people. But when you think about sort of fatality rates and things like that, it has a lower fatality rate compared to some of these other viruses.
Sure. So being in the same family, if you know enough about one, how does it help unlock clues about another one?
Yeah, I mean, there are things that you can learn, and so you definitely use the most related virus to take a first guess of how this virus might behave, or might present itself. But often we can be wrong, like with SARS-CoV-2, because we're learning about this virus every day, and we're seeing this really wide range of disease and symptoms and severity, and it's not like anything we've seen before. And so just because you have a SARS-CoV-1 that's closely related to this, it doesn't mean that it's going to sort of be the same or very similar disease. It can surprise you also. But there are things that we can learn, that are similar as well.
Right, right.
Yeah.
Colin, let's try again on the symptoms thing. How can we do a test and see if we can hear you now?
Yeah, is that any better?
Oh, it's fabulous. Yeah.
Okay, good.
Yes. I was
So,
afraid we were going to have you shouting into the screen. So no, we can hear you now. So that's
okay, good. So coronavirus mainly enters through the respiratory tract, so the lungs, but that would mean entrance through the nose or through the oropharynx. However, it is a syndrome, meaning that it has a constellation of different symptoms, and probably the most prominent one would be pneumonia, viral pneumonia. However, this virus has been very odd in its behavior. There's been viral disease found in people's hearts. There have been coagulation cascades that have caused people to develop deep vein thrombosis in their legs or pulmonary embolisms. And there's also been strokes that have been associated with this virus as well. And this isn't necessarily in populations that we would think of for strokes, kind of older individuals. This might be in people in their 30s and 40s. So it's been a very odd disease and has a lot of different components. And we also see that the residual effects from the disease can last for weeks, months. I don't think that we even know what the overall sequelae will be for people who have been infected and still have ongoing symptoms, whether or not that they will persist, kind of indefinitely.
Mhm. This is a really crude metaphor, but it seems like whack-a-mole to me, that we figure out one thing that it does, and then three other things pop up in terms of symptoms and how it manifests in different people.
Yeah, and we don't really have a good way of predicting who is and is not going to have major complications. There's many people walking around who are completely asymptomatic.
Which is kind of the scary part, right? I mean, if you don't even know you have it, how would you know that you could spread it, right?
Yeah, exactly.
Okay, great. I want to go back to the genetics part of it. Pavitra, on the genetics of the virus, how does understanding the genetics help us understand and develop treatments or vaccines or a way to deal with the vaccine? What does that information provide us?
Yeah. So when we talk about genetics, I want to divide that into like the viral genetics part of it as well as the host genetics, because they're both different and very important. So when we're talking about the virus's genetics, what we're talking about is what its genomic sequences. That's the sequence of letters that encode all the proteins that make up this virus. And so when we try to determine that sequence, we're doing that because that gives us a lot of information about the different parts that make up the virus. So one thing that the sequences can tell us is, of course, how closely linked viruses from two people who got infected are, and that can tell us whether or not that they might have gotten infected from the same source. So that's the public health angle of things, determining where these clusters of or these outbreaks are. So genomics, the sequences of the virus can tell us that, but also crucially, we want to keep an eye on mutations that arise in this virus's genome over time. So this is something that's going to happen. It's not, like the science fiction movies where it's like, oh my God, it's mutating and like that. But it's part of the viral life cycle to change and have errors that are mutations that accumulate over time. The key question is how these mutations are affecting the way the virus is making people sick, right? Is it making it more infectious? Is it making it more virulent or causing more severe disease? And the only way we can keep an eye on that is by sequencing the virus and then trying to correlate the sequence with these various different outcomes. So you may have seen the headlines about this mutation, this D614 gene, the spike protein that may have been associated with becoming more infectious. So this is something that arose, or we learned about it because we had these sequences and data from all over the world, and we could see that this mutation was becoming very common, and there was thought that it could be making people, it could be making people more sick, but several different studies have then looked at that, found no relation with the clinical outcome, but certainly we do see that this is an example of a mutation that allows this virus to make more copies of itself when it does infect somebody. So this is just an example, but this is what the genetics or the genome sequence of the virus can tell us. And then of course, there's a host angle. You can also learn things about, there are questions, and other groups are looking at this, that are there particular genotypes in people? Are there mutations in people that make them more susceptible to disease or have severe disease? That's something that's ongoing, and different groups look at that too.
Colin, I wanted to ask you about one thing that struck me through this whole thing is just the amount of information that's coming out and the differences in the information sometimes, and how the information is accepted. As a physician yourself, how do you sort through what you hear, what you read, what you see, and choose a path?
Well, I mean, I think that I have the benefit of working for a pretty large organization. So, Kaiser Permanente has multiple regions. And so they've had a national COVID task force, and then they've had regional task forces. So I think that the task forces are being led by infectious disease, and Dr. Paul Thottengall, who is the infectious disease specialist here, and one of the infectious disease specialists here in Washington, is the national Kaiser Permanente COVID task force lead. So, it really, I don't necessarily have time to sort through everything that's coming out, but I would rely heavily upon the organization that I work for and their desire to have evidence-based medicine as their overall practice. So they have meetings. Initially, they were weekly to disseminate information to the medical group. And now they've spaced out to every other week. But I think that as we see this surge, that they will be increasing in frequency again. So that's mainly how I'm getting the information as it's been changing.
And we're coming into flu season, so how are you going to be, how will physicians differentiate between the symptoms of COVID and the symptoms of a flu, and is it just testing based?
The symptoms are too similar. I think that what they will end up doing is just doing co-testing because they're essentially the same type of swab that are used to differentiate coronavirus and influenza. So unfortunately, there wouldn't be a very good clinical way of distinguishing between the two.
Speaking of tests, how accurate are they? And either one of you weigh in on this. I mean, can there be false positives? Can there be false negatives? From where you're sitting, what's the efficacy of the testing that we're doing?
Yeah. So when we talk about tests, there are actually a wide range of tests that are now available. So I'm going to focus just on the tests that detect the virus. So these are looking for whether or not you have viral material in your body. And even within that category of tests, so I'm distinguishing that from antigen tests, which tell you whether you may, from antibody tests, which tell you whether you had it in the past. So when we're looking at tests for current infection, there are the rapid tests that you can get in the outpatient setting. And then there's the PCR or polymerase chain reaction based tests that you get in a clinical lab like ours. So the PCR based tests work by detecting the genomic material of the virus, and they are the current gold standard, and they are highly sensitive, and they can really find very low levels of viral material. So the chances of, so I guess the false negative with that is it's highly unlikely to get false negatives with that test. The rapid tests tend to be a bit more variable. But certainly with the PCR tests, accuracy is less of a concern. The issue is twofold. One is, are you getting the test too early before you have enough virus in your body to find it? So that's one example of a case where you may show up as a negative, but you may come positive later. So there's that concern with these, even with the PCR tests. And the second question is, when you test positive, are you actually still infectious to other people? That's the other issue. And, just because, so that is related to how much virus you have circulating in your body. So at certain low levels, it's thought that you may not be as infectious. So I guess the answer to your question is, it depends. And it's
like the case of a lot of COVID questions. How about you, Colin? What's your, in the testing that you've seen, what's your experience?
I don't think I can add anything more than Pavitra already said.
Okay. One thing that's been interesting on the U of I campus has been that the research correlating wastewater testing with specific hotspots in housing. And I think that's going to be an emerging field of study for a lot of scientists here anyway.
Yeah, I didn't realize that was ongoing, but that would be super cool. I know that other campuses have done that, and that data certainly shows that you might be able to predict clusters of cases based on testing of sludge and wastewater.
Yeah, it was really helpful on identifying hotspots in the residence halls and Greek housing. So yes, more to come on that.
That's one of the, I mean, this isn't necessarily COVID related, but as certain infectious diseases become more and more uncommon, that is a way that some surveillance can be done for them by checking wastewater. And for an example of that has been looking for polio. Since polio has become so infrequent, they can find polio within wastewater treatment to see if there are outbreaks within certain parts of the world. And there is surveillance that's done for that.
Yeah, it's, I think with every new virus, new thing that comes there will be research surrounding, taking a closer look at it. So we have to talk about vaccines. There's been some amazing news this past week that not just one, but there may be two vaccines that are moving forward at a pace that might promise something early next year. I've heard by the end of this year, but it's November, so I'm just thinking early next year. Colin, what does that look like, and what are the issues surrounding any sort of vaccine development and distribution? So,
I mean, I think that this will be an interesting story that there will be a lot of books written about, I'm sure, in the future. But just to give a little bit of my background, I worked for the Kaiser Permanente Health Research Institute in 2013 for a pandemic preparedness study that was based on a avian influenza that was a novel influenza circulating in China at that time.
Mm-hmm.
So when the picture of this pandemic started to show itself in January, I reached out to the Research Institute again to say, hey, it looks like you might need some help. And just, they had just submitted an application for the phase 1 study of the Moderna vaccine that was in the news yesterday.
Wow.
So they had been working on that application even as early as the last week of January. So the Moderna vaccine itself was produced off of the viral genome that was released from China in January, early in January. So the vaccine itself was developed off of the genome, and it wasn't developed off of any actual living virus. It was just looking at, what the genetic material was, and then what the sequence of mRNA that would be needed to produce that same genetic material within human cells. And so they were awarded the phase 1 study, I believe, the first week of February. And then they started recruitment for that study at the end of February.
Mhm.
And then their first vaccine was delivered in the clinic that I work at. The first vaccine to a human subject was given 66 days after the release of that genetic sequence from China, which is really extraordinary. I think that it's hard to emphasize how unbelievable that is. That type of research generally takes years to be done, and to have it done in 66 days was really unbelievable. So that phase 1 study enrolled the first human subjects over the course of March and early April. And then the first results of that study were released in July in the New England Journal of Medicine. That study had really good safety and was well tolerated. And so they expanded the study to include people over age 55. So the study was then expanded to enroll a number of people in their 60s, 70s, and 80s. And that study was then released in the New England Journal of Medicine later on this fall. So Moderna then moved on to doing their phase 2 study in May and June, and then their phase 3 study started enrollment in July.
Okay.
And in that time period, they've enrolled over 30,000 subjects across the United States, with 15,000 in the active group and 15,000 in the placebo group. And that's the results that were in the news yesterday. So, you know, the studies that have been done so far right now, the New York Times has a really great dashboard about all of the different vaccines that are being investigated across the world. The two that are in the news right now are both mRNA vaccines. There are also some adenovirus-based vaccines that are being researched that I think will also be in the news relatively soon, as they're in phase 3 studies as well.
I don't know what an adenovirus is.
Adenovirus is a type of virus that causes like common cold, but there's different types of adenoviruses, and they can actually use an adenovirus that affects mostly chimpanzees, and they can use that as a way of getting the viral material inside the human cell, but not get the human subject sick from the adenovirus, since it's not able to continue replication.
Okay.
And so it's just another way of having a vector to get the genetic material inside the cell.
Okay.
So there's about 12 phase 3 studies going on across the world at this moment. Phase 3 studies are the ones that lead to ultimately the safety and efficacy data that is needed to then submit for approval of a vaccination.
Mhm.
So right now we're seeing that there have been 2 that are at the point in the United States where they will be able to submit for emergency use in the next weeks.
Mhm.
And I think that the big differences between the Pfizer vaccine and the Moderna vaccine is really their storage temperature. The Pfizer vaccine needs to be stored at negative 70 degrees, and it's going to be very hard to disseminate that type of vaccine. I think that the Moderna vaccine in many ways has a leg up because it needs to be stored at negative 20 degrees.
Mhm.
Negative 20 degrees is frequently what clinical locations have in there as their freezers, because
So
the
they're
varicella
already set for that.
vaccine needs to be stored at negative 20 degrees as well. So, varicella being chickenpox, that's a vaccine that's given at basically every primary care clinic, pediatric clinic, internal medicine clinic. So I think that being able to disseminate that vaccine is ultimately going to be easier than the Pfizer vaccine, but it's great that there are potentially 2 vaccines with extraordinary efficacy that are in the pipeline as of the last 2 weeks.
Do you think that time frame is realistic? The headlines have said by the end of the year or early next year.
It looks like the FDA will be looking at this in the next weeks in terms of the Pfizer vaccine. I think that Moderna probably won't be too far behind them with submitting their safety data. And then it will need to go to the Advisory Committee on Immunization Practices, which is an independent committee outside of the FDA that will give their recommendations as to who should get the vaccine first and what if there are any particular safety concerns that they have. But I do think that these companies have already been manufacturing vaccine and that there would very likely be cases or vaccine that would be disseminated by the end of December in certain groups.
It really is amazing when you think of the amount of science that has happened in such a short amount of time with these results. It is borderline miraculous. It's very impressive.
Yeah, again, I think it's hard to emphasize it more.
Yeah.
These types of vaccines generally take many years to go through all of their studies. And I think that it's really important, especially in this context, to discuss that there haven't been any corners cut on these studies. They have been, they're the same studies that any vaccine would need to go through in terms of safety and efficacy. The one difference being is that they were done contiguously. Phase 1 happened. Once there was enough safety data, they moved to phase 2. Once there was enough safety data, they moved to phase 3.
Sure.
Whereas usually a lot of these studies are done phase 1, wait, phase 2, wait, phase 3, wait. So that's the part that's different. But we haven't done any thing that would be outside of a normal study for these studies.
I think that's really reassuring to a lot of folks to know that the processes have been followed, even if it's been in a speeded up fashion. We have a lot of questions from our viewers, so I'm going to turn it over to Christy, and we'll try to get through as many as we can. Christy?
Okay, as you can imagine, we have a lot of questions, so we'll try. I'm going to just say them in the order that they were received. So, how many different COVID-19 serotypes have been identified?
1.
Okay.
So there's just there are no like strains or serotypes yet. What we have are what we call clades, which are sort of groupings of viral sequences that look similar to each other. So a number of different clades have been identified, but there is only 1 strain so far, because it's important to remember that when you talk about strains, there's a very specific definition of what a strain is, and we don't have different strains yet.
Okay, and that kind of speaks to the next question, and maybe it's worded incorrectly, but could you explain the difference between the 2 strains of the virus from Wuhan, West Coast, versus the virus that mutated in Europe?
Right. I think that's probably referring to that D614G mutation. So yeah, so it's not technically not a strain. It's not defined as a strain. We call it a variant technically. So the G variant, like I sort of briefly mentioned earlier, it differs at a specific position in the genome within an area called the spike protein, which is the little protein that sticks out of the virus that the body recognizes. And it's this mutation that sort of changes that protein a little bit. And that is what is causing the differences in how it sort of behaves, in the sense that it's been associated with people who have the G variant tend to have more virus in their nasal swabs compared to people who have the D variant. So that's the main difference, the main reproducible difference that we've found in those 2 variants so far.
Okay.
And we keep an eye on these new variants as they arise.
Okay, Sandy has a couple of questions that she's going to ask.
Kind of segued into the question that I got was, we hear that the virus may have come from a wet market in China, human exposure to exotic animals, or even a lab in China. Do experts know where it came from and if we'll ever know for sure?
Colin, do you want to take that? So
I don't think I have a good answer to that.
yeah, that's where it was certainly reported. That was the place where the outbreak was first reported among, you know, in a wet market in China, and that's what we know so far. And yeah, so we don't know the exact jump to humans and where exactly it occurred and things like that, and that's still a hotly debated subject as well. So.
Okay. Okay, is herd immunity even a valid option in the case of the US, or would that entail far too many casualties?
Go ahead, Colin.
I mean, I think if we don't do anything, herd immunity could happen, but it would require probably millions of deaths. I mean, I think the issue with herd immunity is that we are seeing that there are people who have gotten reinfected. Just because you have clearance of a natural infection doesn't mean that your immunity will persist indefinitely. And I think that we you know, we just don't have enough time right now. I think about right now, we're getting to probably the time that this virus first started circulating a year ago. And so we won't really even know in the people who were infected 1 year ago as to what their levels of antibodies are now until maybe January. And so if we're not able to have persistent immunity, then herd immunity isn't really an option. I think that one of the things that's really important to emphasize that we've seen in some of the vaccine studies is that the vaccines have actually produced higher levels of antibodies than natural infection. And that's a really important thing for making sure that the vaccine might last long enough to be effective and to get us to a point where we would have herd immunity through through vaccination. So I think that that's one of the main benefits of this vaccine is that we're actually seeing higher levels of immunity than natural infection.
Okay.
Yeah, I think it's important to distinguish if we're talking about herd immunity by vaccination or herd immunity just by allowing COVID to spread, you know, through the population.
I think it's what they were referring to, but I'm not sure.
Yeah, so if you're talking about developing immunity by just allowing, you know, the infection to sort of spread through, I think that the number of deaths would be just so staggeringly high that I'm not sure that anyone would be okay with that scenario. But if you're talking about, but we can achieve herd immunity via widespread vaccination. So it's really important to have high adoption of the vaccines when they come out. And hopefully that will be the case. And hopefully all the safety data will be reassuring to people so that they would want to take the vaccine, because that's a safer way of achieving immunity and sort of stamping the virus out. So that would be a preferred way to go, I'm sure, for all of us. And
I think that one of the areas too, when we're talking about herd immunity in relation to natural infection, is that the healthcare system could not possibly deal with the level of patient need. There would be, you know, inevitably some type of collapse within the medical field if all doctors and nurses are getting infected too. The other thing that I think isn't talked about when it comes to herd immunity is, you know, how much comorbidity are we willing to accept too? You know, we are seeing people having very debilitating conditions come as a result of natural infection that aren't necessarily death. You know, that includes strokes or heart disease or chronic lung disease. So we would have basically the entire population with a pre-existing condition at this point in time after natural herd immunity.
Right. And let's not forget that we are identifying people who are getting sick again. So I just realized that Cassandra, a fellow Vandal, is also on this call, and she's at the Department of Health. And I know that they're keeping a close eye on people who get infected again and again. So.
Okay, there's a lot of questions about vaccines. So will the vaccines currently in clinical trial be safe for immunocompromised patients? And do they carry the danger of enhancing the infectivity of other serotypes?
Well, I think that we would have to look at different categories of immunocompromised individuals. All of these studies have different protocols, so I wouldn't be able to explicitly comment upon each protocol because I'm not involved with all of them. However, a lot of times the studies don't allow for, immunosuppressant drugs like chronic use of steroids or methotrexate or immune modulators. So I don't think we have a good answer to that particular question. However, some of the studies do have other groups of immunocompromised individuals. So people who have had chemotherapy in the past, people who are living with HIV on antiviral medications, they are, if stable, able to be enrolled in these clinical trials. And so there are people who have these conditions who have been subjects in these studies. But I don't think I have more of an answer than that.
Okay, this one is for Pavitra. Are the genomes being sequenced in the positive cases that of the host or the virus?
The virus.
It's quick and easy. Typical med and vaccine development takes years and years. How will the accelerated nature of COVID vaccine influence future research and development of medications and vaccines in general?
I think we'd already talked about how there are no corners being cut in this process of development. So there's that. But it's important to note that a lot of the things that we are doing quickly now, or able to do quickly now, have been from lessons we've learned from other diseases like HIV. For example, the vaccine trial network that is in place right now is based off of an HIV, a large similar trial network that was developed for HIV here, and across the world. So there are lessons that we're using already from other viruses to help us with this one. And similarly, I think there will be lessons here that we can apply to future outbreaks. Just one example is that I think there's going to be an increased interest in genomic surveillance to look for these kinds of novel pathogens hopefully by routinely sequencing to find these kinds of things. There's a lot of emphasis on data sharing. I think the networks that we've built and the infrastructure that has been built because of COVID is going to be super helpful for future outbreaks. Hopefully there won't be too many more.
I think that the two vaccines that we're looking at right now between Pfizer and Moderna are both mRNA vaccines. There are not any mRNA vaccines approved for use in humans at this point in time. However, I do think that type of technology could end up lending itself well to vaccinations for other viruses that we are seeing circulating. And there may end up being some very interesting research that's able to be done when there isn't such a huge, enormous international public health crisis.
Mm-hmm.
And let things calm down a little bit and then you can explore other things.
Okay, Sandy has a quick question.
Will you speak to, and you kind of did a little bit, will you speak to the potential short and long-term side effects given the fast-track nature of the vaccination development?
Well, I think that's a great question, because I think the first thing to emphasize is that the vaccines have been really well tolerated. They have the most common side effects that we've seen, and I wrote them down so that I could tell you all: fatigue, headache, chills, muscle ache, and pain at the injection site. And those are, very common side effects for vaccinations in general, because you do need to stimulate your immune system. And really, the signals that your immune system sends are often the things that end up making you feel ill. But overall, these types of side effects have been very well tolerated and usually don't last more than 1 to 2 days. There have not been, from what I know, any major side effects that have been reported. That there was one side effect in the phase 1 study, but the phase 1 study was looking at different dosages, and that the side effect that was noted was in the highest dose, and that is not what has been ongoing for the further studies. When it comes to long-term side effects, I think that, the jury is still out. So far, of all the subjects who have received the vaccine starting in March and April, they have all been doing very well and haven't reported any new medical conditions. However, we won't have 1 year worth of data on them until March and April. And so I think that at this point in time, there hasn't been anything that's clearly declared itself, but we still, there is still some uncertainty.
This is kind of a follow-up to that one. Once an effective vaccine is available, do we have an idea of how long the protection is likely to persist?
We will have more information about that in March and April, at least for the first subjects. Again, I'll just emphasize that when they looked at natural infection versus vaccination, the vaccination had, more than 1 or 2 logarithms higher development of antibodies, which when you're looking at a logarithm is quite a bit more antibodies. So we will see in March and April, when we draw blood for those subjects who were in the initial study, if they are still having high enough levels of antibody titers.
Mm-hmm.
I think we have time for one more question. The time has just flown by.
This is a good one to end on because it kind of pertains to current students. Where is it? Is there a shortage of research specialists in the area? Do you foresee an increase in students who are interested in biomedical science because of the ubiquitous nature of this virus?
I definitely hope so.
Yeah.
I hope that more people want to study science and want to get involved in this kind of work. There's certainly, in our lab, we've been, like I said, it's been a all hands on deck situation. We've grown, we bought an entire floor to accommodate instruments to do testing. So to go with the instruments, we needed people. So we've hired a ton of people to do that. And we've certainly pulled in techs from all different other labs and volunteers to do specimen processing and all sorts of things. So there's definitely involvement across the board. And yeah, so I think, and there's also, there's other side effects like people wanting to study genomes. So lots of people are interested in bioinformatics, and some of the tools that have been made available, one of my favorite things is how some of those tools are open source. Like there's a tool called NextStrain that you can go to and look at trees of samples that, of sequences from real samples and how they relate to each other. And you can play with that data. So tools like that have made this data really accessible and available. And there's people, that are more interested in it. And I hope that continues. And I hope that, yeah, more people want to do science and code and do math. Yeah.
I think that the globe has gotten a crash course in biomedical research in a way that they never would have paid attention to in the past. I mean, I think that when people are asking about phase 3 studies and what's this and halting criteria, et cetera, it's just something that was always happening in the background, but people didn't, it wasn't a direct effect on their lives. I think as an anecdote, my cousin who lives in Pennsylvania wrote me a message saying that her daughter wants to become a vaccine researcher when she grows up. So I think that there's an awareness in, these younger generations.
Yeah.
And we've actually gotten some very lovely letters from children across the country, just asking us questions and things like that, that I think, they're really paying attention to what's going on around them.
Yeah. Well, if there isn't 2 better alumni for our future scientists, then U of I, you 2 are it.
I agree. So
I just wanted to make sure that we never hit on the question of who was your favorite teacher at University of
Oh,
Idaho?
yes.
Oh, yes.
So
Yeah.
I have to give a shout out to Gary Williams. He was definitely my favorite teacher. But I have to also give a shout out to Gustavo DeVico. I don't know if he's still there or not, but that was the hardest class I ever took, and I think it really prepared me very well for medical school. So
That's great.
That's awesome. How about you, Pavitra?
Yeah, definitely. A big thank you to Steve Crone, who's my major professor at Department of Math. He's not here today, so please tell him I'm very upset.
We'll
I'll
send
get
him
right
a
on that.
recording.
But yeah, he was just wonderful. Just taught me to be sort of independent and do a lot of different things. And also, I have to mention Paul Joyce. He's the one who got me into science communication. I would say he's the one who he drove us to Boise, I think, and he got us to talk to young girls studying STEM and to encourage them to stay in science or to continue to study math and science. So yes,
That sounds like Paul.
I think about him and think about him and his lessons a lot.
Yeah.
That's awesome. That's awesome. Well, what a fascinating conversation, and it's just beginning. I mean, it'll be really interesting. We should visit again in 6
We
months
do. Yeah.
and see what we know then, and what from your both of your perspectives. It's
That would
been
be
great.
really interesting. I just want to mention that I do have some other questions that we weren't able to get to. And I think what I'll do is just email them to Colin and Pavitra, and then I will have them answer them and then get back to the people that asked them. Sorry.
That would be great. Thank you, Christy. Before we end, we have to do our drawing. So, Sandy, do we have a name for our drawing?
We do. It is Mara Fields.
Thank you.
that's my mother.
I was like, we wondered. I was like, I wonder.
yeah.
And it's a random draw that Marie gives us. So we wondered if there was any relation.
That's awesome.
Hey, congratulations, Maura.
So that's awesome.
We'll get you your Vandal store gift card as soon as we can, Mara. So congratulations.
Oh,
Glad
thank you. It was rigged.
It was rigged.
So
right.
again, I just want to say a very sincere thank you to both Colin and Pavitra, two amazing Vandals, just doing, right on the front lines of something that has changed our world. So thank you for that. I also want to thank everyone for attending. I know a lot of you have been with us from the very first episode of Cup of Joe back in August, and thank you for joining us all those times. We're going to pick up again in January with a St. Mary's native, St. Mary's, Idaho native, Tom Mueller, who is a retired executive from SpaceX, and he has a very successful launch to talk about in January. So until then, I wish you all a happy, healthy, restful holiday season. And as always, go Vandals.
Go Vandals.
Thank you.
Thank you.
Thank you very much.